Hi everybody! Welcome again to our channel of Biomol Info. Today we got to you the process of RNA splicing. That is a molecular biology process in which a new mRNA, messenger RNA, is created from a pre-mRNA. This mature RNA consists when the introns that are non-coding regions are removed from the RNA and the exons are kept, and the introns are removed mainly because there are high regions where mutations can occur. This commonly happen in eukaryotic type of cells because there are genes that contain the introns, and splicing is usually required in order to create a messenger RNA molecule that can be translated into a protein. And for many eukaryotic introns, the splicing is carried out in a series of reactions that are catalyzed by an enzyme called spliceosome. Really difficult to say, and it's a complex, a small nuclear ribonucleoprotein.
Several methods of RNA splicing occur in nature. The type of splicing depends on the structure of the spliced intron and the catalysis required of splicing to occur. The first one is the spliceomal complex. This is the most complex splicing method, so I'm gonna only explain the superior part. The splicing is catalyzed by the spliceomal, a large RNA protein complex composed of 5 small nuclear ribonucleotides. Assembly and activity of the spliceosome occurs during transcription of the pre-messenger RNA. The RNA components of snRNPs interact with the intron and are involved in catalysis. Two types of spliceomes have been identified, major and minor, which contain different snRNPs. Second one, recursive splicing. In most cases, splicing removes introns and single units from precursor messenger RNA transcripts. However, in some cases, especially in messenger RNA with very long introns, splicing happens in steps, with a part of an intron removed and then the remaining intron is spliced out of the following step. This has been found first in the ultraviothrax or UVX gene of the fruit fly. 1. tRNA splicing. This splicing is another rare form of splicing that usually occurs in tRNA. The splicing reaction involves a different biochemistry than the spliceosomal and self-splicing pathways. The fourth, trans-splicing. Trans-splicing is a form of splicing that removes introns of outrons and joins two exons that are not within the same RNA transcript. Fifth one, the self-splicing. This occurs from rare introns that form a ribozyme, performing the function of the spliceosome by RNA alone. There are three kinds of self-splicing introns: group 1, 2, and 3. Yeah, very original.
This splicing process is taken in two main steps. In the first step, the pre-mRNA is cut at the 5' splice site. The 5' end of the intron then is joined to the branch point within the intron. This generates a latriate-shaped molecule characteristic of the splicing process. And the second step is when the 3' splice is cut, and then the two exons are joined together and the intron is released. Well, sorry, this was a lot of information, but I hope you really get to understand it because this is really important. This represents an important part of the central dogma of molecular biology because without this, proteins cannot be done. And of course, we need proteins to live, and also splicing is really important in the manipulation of genetic information. So really thank you for seeing all our stories. Hope you really get to enjoy this information. Don't forget to follow us, and of course DM us if you've had any question of this team and keep informing yourself in Biomol Info.